Hi, this is Julian Romano, and this is my protein synthesis project. Right here we have the DNA structure. The blue represents guanine, the yellow represents cytosine, the red represents adenine, the green represents thymine, the white represents phosphate group, and the purple represents the deoxyribose. DNA structure. DNA is made up of molecules called nucleotides. A nucleotide is a S compound consisting of a nucleoside linked to any phosphate group. They contain a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA's genetic code. Human DNA has around 3 billion bases, and more than 99% of those bases are the same in all people, according to the U.S. National Library of Medicine. The order of nitrogen bases is a DNA sequence forms genes, which is called the language of the cell. It tells the cell how to make proteins. RNA translates genetic information from DNA into proteins. Nucleotides are then attached together to form two long strands that spiral to create the structure called a double helix. The bases on one strand must pair with the bases on the other. Adenine pairs with thymine and guanine pairs with cytosine. To fit inside the cell, DNA is twisted or collided tightly to form the structures of chromosomes. Humans have 23 pairs of chromosomes, which are found in the cell's nucleus. DNA function. Information encoded in strands of DNA controls the genetic makeup of organisms. The four roles DNA plays are replication, encoded information, mutation, and gene expression. So here we have our hydrogen bond, our lagging strand, which you can see over here, and we have our leading strand. So the leading strand is a little bit longer than the lagging strand. This is the leading strand, this is the lagging strand. And you can see our hydrogen bonds. Now we move on to DNA replication. DNA replication follows several steps that involve multiple proteins called replication enzymes and RNA. DNA replication occurs in the S phase of interphase during the cell cycle. Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. Adenine only pairs with thymine, and cytosine only binds with guanine. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into a Y shape known as replication fork. Right here. DNA is directional in both strands, signed by 5'1" and 3'1" ends. However, the replication fork is bidirectional. One strand is oriented in 3 to 5 direction leading strand, while the other is oriented at 5 to 3 lagging strand. The two sides are therefore replicated with two different processes to accommodate the directional difference. Once the DNA strands have been separated, a short piece of RNA called primer binds to the 3 end of the strand. The primer always binds as the starting point for replication. Okay, the last step is transcription and translation. Transcription and translation. The process in which cells make proteins is called protein synthesis. It consists of two processes, transcription and translation. Transcription takes place in the nucleus and uses DNA as a template to make RNA molecule. RNA then leaves the nucleus and goes to ribosome in the cytoplasm, where the translation occurs. Translation then reads the genetic code mRNA and makes a protein. Transcription is the first part of the central dogma of molecular biology, DNA to RNA. Steps of transcription. Step 1. Initiation. The beginning of transcription occurs when the enzyme RNA polymerase binds to a region of the gene called the promoter. Step 2. Elongation is the addition of nucleotides to the mRNA strand. Step 3. Termination is the ending of the transcription and occurs when RNA polymerase crosses a stop sequence in the gene. See where it's separated, where it's copied. The copy part is then moved away. The separated part then goes back together. And here the copy part is moved and is put into a ribosome. Bring it all together. I'll go over it one more time. So this is the DNA structure. These are the hydrogen bonds. This is the ligand strand, and that is the leading strand. This is DNA replication. It occurs right here in this diagram. And this is transcription and translation. The sources for making this presentation come from ScienceDaily.com, education.seattlepie.com, www.yourgenome.org/facts, www.docco.com/DNA. Thank you for watching my presentation. Hope you enjoyed it. See you next time.